Hi, my name is Anny, and in the summer of 2020, I was a MiCM scholar where I worked with Dr. Marie Kartinen on doing profiling of the matrisome of the adipose tissue in weight discordant twins. So, the adipose tissue is composed of adipocytes, and it stores energy as lipids, and its main role is to regulate whole body energy homeostasis. But in unhealthy weight gain and obesity, the adipose tissue undergoes changes in the adipocytes and in the extracellular matrix, or the ECM. Adipocytes are proliferated and become inflamed, while the ECM remodels itself. And this remodeling is required in adipose tissue expansion, but if it's not regulated properly, it may lead to metabolic failures such as type 2 diabetes. And so for this project, we were interested in the changes of gene expression related to the ECM during weight gain. And more specifically, we wanted to identify differentially expressed ECM genes, or the matrisome, in the adipocytes and the adipose tissue. And lastly, we wanted to identify the roles of these genes in adipose tissue expansion. And so in our experiments, we emulated weight gain by looking at weight discordant monozygotic twins. And more precisely, we compared the gene expression levels of the adipose tissue and adipocyte of the heavy individuals compared to their leaner twin. And this value we called delta. And so the significance was determined using linear model fit and empirical Bayes. And the results were then refined using Benjamini-Hochberg procedure. And the matrisome genes were identified using the Matrisome project, which is an open source database. So with our results of delta values, we generated volcano plots of the whole transcriptome to determine how much of all gene alterations were from the matrisome. And what we found was that the changes in the matrisome makes up approximately 10 to 15% of overall changes of the transcriptome. And then we focused on just the significant matrisome genes and generated a second set of volcano plots in which we identified all significantly differentially expressed matrisome genes. And we also grouped them into the respective categories, which are the types of proteins that are present in the extracellular matrix. So to show the level of significance between all these genes, we generated heat maps for each category to further show the alterations. And what we found was that ECM glycoproteins, ECM regulators, and secreted factors were altered the most in the adipose tissue expansion, while collagen and proteoglycans were less affected. And between the adipose tissue and the adipocyte matrisome, there were 45 significantly differentially expressed matrisome genes that were shared, and we wanted to find the relation between the alterations of these genes and metabolic dysfunctions that come with adipose tissue expansion. So by doing enrichment analysis, we found that the biological processes of upregulated genes were associated with inflammation, while downregulated genes were associated with angiogenesis. And so these findings suggest that gene alterations in the extracellular matrix of adipose tissue may lead to adipose tissue dysfunction. And in conclusion, by looking at the differential gene expressions of the matrisome of weight discordant twins, we were able to profile the extracellular matrix in the adipose tissue. And moreover, we identified genes that may have a role in metabolic dysfunctions caused by remodeling of the adipose tissue. And these results can contribute to understanding the adversive effects of obesity and the root of health risk related to chronic weight gain.